Just want to let you know, my teaching style, I like to write on the blackboard a lot. So you can take notes. You also can take notes with you on the side of your slides, okay, electronically or handwriting, both are fine, but that will be my suggestion. Okay, first question, what is microbiology? So we have microbiology. So what is that? I will say this is science of microorganism. Okay, we'll get to the detail of microorganism later on. So what is the science of microorganism? Basically, we'll talk about numeration, identification, that we'll talk during the semester. We'll talk about some bacteria caused disease. And then we will talk about biochemistry plus some of the genetics. But this will be very basic, which means different from the biochemistry class. You don't have to remember, you don't have to write all the chemicals. We are going to be a general education, that's what we call it. Then we're going to talk about is applied microbiology. Applied microbiology, basically this is lots of fun parts. So like poultry meat processing, antimicrobials, all those things, okay? So that's a science of microorganism. Now this is talk about what is microbiology. It's concerned with study of microorganisms, including algae, bacteria, virus, protozoa, and fungi. This is an important slide. Okay, what is microorganism? So I took this off. We want to talk about microorganism. So we have microorganism. So microorganism, this is a word I usually split them into two. This part is micro. What this means? Too small to see by naked eyes. You cannot use naked eyes to see most of the microorganisms. There is an exception, is fungi. Some of the fungi you can see it, so there's a debate about that. Okay, with some exception, but most likely it's too small to see by naked eyes. We need to rely on a tool, the tool called microscope. So we say rely on microscope. Okay, what is organism? This is a live body. Okay, this comes out with a big question. What are the examples of microorganisms? You can read the slides. It's messy, lots of words there. You pretty much don't like it. So let's make your life easy. Let's talk about something. Everybody knows microorganisms. There is one type of the microorganism everybody heard in the last 3 or 4 years. What is that? Virus. Is that right? We had COVID-19. So what's that? Coronavirus ID 19. So that's virus. Okay, what else? Rabies virus. We call it a crazy dog virus. Okay? HIV virus. That's a virus. So what's a virus looks like? If you want to look at the virus structure, it's very simple. Like that. The outside is protein. Inside is DNA or RNA, but never both. And then you have so many like a crown there. It's like a crown. That's called a coronavirus. That's a very simple structure. That's a virus. What are the second example of a microorganism? Everybody heard. Bacteria. Is that right? Did you heard about E. coli? I often make a joke. I said, you don't know any bacteria, you don't know any microorganism, but you absolutely heard is E. coli. Did you heard of salmonella? We always have outbreaks of salmonella in chicken products. What else have you heard about? Listeria sometimes. So that's bacteria. Okay, what else? Fungi. Fungi have two categories, yeast and molds.
I forgot something. Sorry, Dr. Shan.
That's okay.
I can't do any work without this. Thank you.
There are different growth environments. So yeast, 37 degrees Celsius. And molds, usually room temperature, 25 degrees Celsius. And you know about molds. So you have a bread, and you have a couple bites. So you put in your dough, put on the table. And you don't touch it for a couple weeks. So what you see? You see the greenish color on the surface. What's that? That's molds. And in your house, if it's a very rainy day or like continual raining like a couple weeks, so you see the ceiling start to have a dripping, so water drops, and then later on it become like a greenish or blackish color. That is molds. Okay, so that is fungi. What else we have? Protozoan. Okay, protozoan, what we have? We have the one which is relatively large size compared to bacteria and fungi, but they all rely on host. So for example, Plasmodium. And we say that the pregnant woman has to be avoid have cats at home because Toxoplasma could be cause miscarriage. So that's protozoan. These are the major things. And all these three, we say cell is a single unit, so we call it a cellular. And the virus is non-cellular, so we call it acellular. Okay, so bacteria, fungi, protozoan, and the virus are examples. What are the others? You go look at the slides. We talk about fungi, yeast and molds. We talk about bacteria. We talk about algae, protozoan, slime molds. So we're going to explain something to you. What is slime modes? Yes.
Oomycetes, right? They're just a single cell, like just one
Yes,
cell.
single cell. Cellular, which means a single cell. Acellular, which means not a single
I
cell.
mean,
It's a
like,
unit.
even just the big organism.
Yes.
Is just all one.
That is a very good question. There's a debate about this definition. We'll talk about this using naked eye is too small to see. But actually, some of microorganisms, you can see it. So that's in the older time, we have a question. After this class, can you give a definition of a microorganism by yourself? That is always a very good question. Think about that. I mean, this definition covers 90% of the bacteria, we can say, but not for the other things. Okay? Very good question. Slime modes, it's a little bit tricky. It's a modes, so it should belong into fungi. But it's a protists. Why? They have a two phase, which is like a fungi and like a protozoa. What they do, they could engulf the leafies and the debris in the environment and use them. So we also call it a natural cleaner. Okay, that's slime modes. Messagens, what's that? Messagens, this is something you know. You learn chemistry? Is that right? This, we have one. So what happened is this guy here, you have, that's very interesting. Sometimes I like to draw these because it's interesting. You have this guy right here, this guy right here, this guy right here, this guy right here. That called Lewis structure. So what is this? Meson. Look at the two words, messages. That's archaea, can generate a meson. In the older time, this is going through fermentation, they can generate energy for electricity. And where they come from? Horrible. Human fecal samples going through fermentation. So that's called archaea. Okay? For others, we mentioned about virus. What is varroids? Varroids is usually only composed by AEM. And it is a plant pathogen. In 1996, it has been damaged by a huge area in the Philippines for those palm trees. So it's a plant pathogen. Next one, satellites. You don't have to worry about that because I never heard about it, which barely mentioned. So you go back to look at the structure. We talk about the DNA or RNA. We talk about the protein. They can compose, become a virus. If they are composed with a cell wall, then with carbohydrates, with amino acids, or other things combined together will become a bacteria, fungi, protozoa. But how about this only protein? It is a protein from a chemical standpoint, but it is also a microorganism. What's that? Prions. Did you ever heard mad cow disease? That's what happened. Started in 1997 in Scotland, in Great Britain. So those cows have like neurological symptoms. They are untreatable. The only thing what you can do is burning them and debury. And those microorganisms could be contaminated with human being. Human will have a colo or scripts or those things. And then it started to do the international exchange or the contamination that go to the United States. So start from 2003, the first case of the medical disease found in the United States, which caused the damage of the meat industry. So even today, you cannot ship the meat products to Australia, to Russia, and to China. So for example, if you want to buy a beef jerky, if I buy a beef jerky and I mail it or ship it to my friends in China, it will be blocked by the customs. It will not go through because based on this, the 2003 outbreaks. So that's a medical disease called prions. It's a protein. So this one gives you an overview of what is microorganism. So we mentioned about so many, and we can talk about more. But in this class, we will mainly focus on is for bacteria. Because the time is limited, and for the others, we will mention some, but not focus on all of them. Because if we talk about all of these microorganisms, that's another class called the biological diversity class, or microbiology diversity class. So that's another one. Just want to let you know. We're mainly going to focus on the bacteria. Okay. Microorganism is always bad. That's not a problem. Everybody knows. So E. coli. Diarrhea causes urinary tract infection. The number one urinary tract infection is E. coli. Salmonella causes food poisoning. Virus, coronavirus, flu virus, hepatitis. That's always bad, causes disease. So here is something we want to talk about this E. coli. It's very interesting. So this E. coli. Whenever we talk about E. coli, everybody said it's a sickness. It's not pathogenic. Yes? For example, E. coli 0157H7. And you heard about the news. Sometimes CNN breaking news, there is 5 million pounds of the beef at some plants in some states is recalled with is because of the possible E. coli 0157H7 contamination. Yes, that's a pathogen because it will generate Shigella toxin and will generate some diarrhea, vomiting, and nausea. But the E. coli sometimes is also a normal bacteria. We call it a generic E. coli. And generic E. coli could be used to testing fecal contamination. Which is coming to human poops. So for example, if I get a pond water from Cheat Lake, and I go ahead to do the sample, and I find the generic E. coli, what means? The Cheat Lake is contaminated with animal or human poop samples. So that's generic E. coli. So you also know that in the United States, you open your tap water, turn on tap water, it's drinkable. So why? Because EPA have a regulation. It is zero tolerance of generic E. coli, also coliform, in municipal drinking water. So that's generic E. coli. And we say that E. coli could cause urinary tract infection. This is called urological E. coli. And Woman is major risks for urinary tract infection. So that's urinary-logical E. coli. Originally, it's a human pathogen, but later on, it's through the food processing system. It's already going to the food environments, manufacturing environments. But also, I want to mention, E. coli could be a good one. E. coli 9037. This is used for fermentation. Which means it could be a beneficial bacteria. So just want to let you know, even the same name, based on their different species, could be bad, could be good. Okay, so that's the first one. Bacteria cause deleterious impacts. There's more examples. Fish, you see the bacterial infection? Which bacteria will cause the infection in the fish? Vibrio. Conjunctiveness of the eyes. And be careful about that. This is not really only we talk about is the skin disease or the inflammation of the eyes. It could be a sexual transmitted disease showed up with the conjunctiveness of the eye. Tomato blossom and blotted, caused by awonia. It's a plant pathogen. This is everybody knows. Strep throat, caused by a pathogen we will mention. Streptococcus pyogenes. I tell you one thing, if I said you need attending for the class, sometimes you will send me the email saying, Dr. Sheng, I'm not going to be here tomorrow because I have a strep throat. That's a major excuse before COVID-19 comes for not attending my class, in addition to like cough broken or those things. But then you know which pathogen will cause. Is Streptococcus pyogenes. What's a Streptococcus looks like? Like a chain shape. And it's very easy to be diagnosed. When you go to see a doctor, you're coughing, sneezing, he get a cotton swab, go to your nose. I have a rapid test kit and just dump in there, dip in there, showing positive in like 30 seconds. If you have a strep throat, what you need to do? They'll give you the prescription of the antibiotics immediately. Is that right? If it's not, then they will think it's a virus. They cannot do anything. Just go home. Go to the countertop of CVS, buy some of the prescription drugs. That's all. But if you have a strep throat, you need to use antibiotics. Then what happened? Antibiotics we used too much become antibiotic resistant, related to all plasmids. That's the thing. We will talk. Okay? That's a good one. Everybody like it. The beneficial bacteria. Do you like drink beer? Yes, we all like it. Two kinds of the beer we have, ale and lager. Is that right? So when it's top fermentation, when it's bottom fermentation. A little bit dark when it's light. You like wine? Where the wine comes from? Grapes. Where is the grapes doing the fermentation? Saccharomyces cerevisiae, and those are yeasts going through the fermentation. So the red wine, the white wine, what a major difference? It depends whether the grape is crushed or not. Antibiotics we just mentioned. It is a metabolic compounds from one microorganism and kill or inhibit another microorganism. Insecticides used to killing insecticides. So these are usually we using for the pharmacy and the commercial. This is what you really like. Cheese. So we will talk about you like this guy. See those holes? Swiss cheese. Right? Cheese, we like it. So that's yellow color. You see those holes? Carbon dioxide, where they come from? Fermentation. And yogurt, sour cream, they containing beneficial bacteria. What is the major name of a beneficial bacteria? Lactic acid bacteria. Okay, that's a good one. Okay, the next topic, what we want to talk, is vaccination. I usually talk about that because we're just going through the COVID-19. We relate to the vaccination. We've been talking this all the time. For many years, we talk about the vaccination. So we want to introduce you what is vaccination. Okay, so I don't have much space here, so we're going to move my camera. Is go the other side because I can draw on the other side. Talk about the vaccination. It's a very important topic. We can talk about that. So vaccination. V-A-C-C-I-N-A-T-I-O-N. What does V-A-C-C-I means? This means a cow. Latin words cow. So, how do they find it? This is coming from a great British scientist, Edward Jenner. He works as a physician in the royal community in the Great Britain. So what happened? By the time smallpox is a damage in the whole community, because millions of people, trillions of people died, because they will be destroy the immune system. Let's say the bone marrow system. But what he find it's very interesting. The ladies or women working in a cow plant or in the dairy area, they will have those lian showing up, have a very mild symptom. Instead of they have a smallpox, they have a cowpox. Although they still have a pox, but this is mild symptoms, will not cause them to die. So what he did, he got the lian section, generate some of the vaccine, and this vaccine used to inject a boy in the royal community of the Great Britain. Yes?
Were the lesions just like the scabs from the smallpox?
Oh yes. This is usually is a dead white blood cell. So there's a liquid that comes out, exudatives comes out, extracted comes out. Then he gets those, of course he did some purification. Then he get injected to the boys. And then he contaminated the boy with a smallpox virus, and the boy survived. That's why today we want memorization. Edward Jenner, as a great medical scientist, a great physician, we call the process called a vaccination. That's what vaccination comes out. But when you read the textbooks, it always tells you, don't repeat this. Okay? It's unethical to do today, but it's fine. During the development of the science area, that always happens with these type of. So that's vaccination. Now we're gonna jump in, just to let you know some of the basic knowledge. So what's a vaccine talks about? So vaccination is stimulate immune response in a body. Basically, the immune response coming from low dose of toxin. So which means you can understand is that you injected with some toxin to stimulate the immune reaction in your body and generate antibody. So this immune response, you can say it is an antigen-antibody reaction. So that is called vaccination. Because more or less there is a toxin there, so there is a debate. Is that right? They always have a debate whether we should have a vaccine or not. And it is also belonging to the category is artificial active immune. So this topic we want to mention, we can spend your time to talk, but just be very briefly, okay? Related to the immune, there are different categories. So we have passive immune, and we have active. And then we have passive immune. For example, if you are recovered from Streptococcus pyogenes, let's say, this is the passive immune. An active immune, vaccination is one of the examples. Because they'll get a shot, that's an active action for them. But they can be dig more to different categories, like a natural acquired passive immune, like a natural acquired artificial immune. Okay, there are different things. Sorry, this is passive, should be artificial. Sorry. So what we can do the category, okay? We can have a natural acquired passive immune. Then we have a natural acquired artificial immune, active immune. Then we have artificial acquired passive immune. And then we will want to have artificial acquired active immune. So this can dig a hole and talk about an hour. Just give you some example. Natural acquired passive immune, what is that? Recovered from a disease. What is natural acquired active immune? Mother to newborn. What is artificial acquired passive immune? This is some antitoxin. So for example, if you get a Clostridium perfringens, a Clostridium botulism, what happens is canned food. So let's say you eat a canned food, then the spore generates. And what they have to do? Report to CDC. CDC will let Atlanta Airport to shipping anti-equine, antitoxin. This is artificial acquired passive immune. But what is artificial acquired active immune? Vaccination. So just want to let you know, it's a little bit complicated, but basically you need to know passive and artificial vaccination, what is that? This is the one we mentioned about the vaccine could be coming from different resource. It could be the killed microbes, which means microbes has been heat injured. We heat it to let it die. The example is polio and whooping cough. That used widely. But the most effective immune, the vaccination, is attenuated one. This is live cell, but low dose. A very good example, MMR. Mumps, measles, and rubella. Rubella. MMR is required for every immigrant to go to the United States. I gave shots when I studied here 20 years ago. It's most effective. 1 shot in your lifetime. Because it's active both use T cell and B cell. And the longer standing, very good memorization period. Purified antigens, the next one. What this means? Certain structure of the bacteria cell or virus, we took it out, we manufactured, and it become a vaccine. So the examples, meningitis, whooping cough. But it's not very long-standing, so you need a booster. So next one, toxoid. This is a guy. Always we talk about is toxic-like chemicals. What is the one? DTAP. This is a guy. You look at your immune recording, you definitely have a shots of DTAP. What are those 3 things? Is diphtheria, corynebacterial diphtheria. What is T? Clostridium tetanus. What is P? Bordetella pertussis. Is for whooping cough. DTAP, the vaccine, you heard about that. But unfortunately, in your lifetime, you need to have a multiple shots. We call it a boosters. The reason is this is only activate T cell. Okay, generically engineered hepatitis. That's an example. That's why the hepatitis vaccine is always expensive. But don't forget, we have a COVID-19 virus, and the COVID-19 vaccine, where it comes from? The S protein, is that right? From the virus cell. So that's a new developed technology. Yes?
That video that you're recording, are we going to be able to see that later?
Yes, of course.
Okay, I was about to say,
We'll be on the
why-
e-campus.
Yeah.
We'll be put the link there, then you can see it.
Okay. I appreciate
That's why I do it. They always, you can go back to read when you have time. I did it for all my classes since the COVID-19 period. So there's a time, and a lot of people like it. Okay, because you can always go back to read.
Right.
Okay, very good. So that's vaccination process. Okay. So let's see something else. What we have. Okay, the discovery of microorganism. This one we don't need to draw on the blackboard. We can just describe here. If you can read the slides, we want to talk about that. So who is the first person to discover microorganism? Is Anthony van Leeuwenhoek. He is the person from Netherlands, and he is an amateur scientist. He always says that because he does the business It's traded all the time in the European countries. He had a very long life, you can see that. He is the first person to observe and describe and record microorganisms accurately. And based on a very magic tool. It is a mechanic microscope. So you see there's a lens, there is a specimen holder, there is a handle, there is a cross focus screw. What is the magnification of this microscope? I tell you one thing, it's very limited. It's only 200. What's the microscope we use in the lab? Around 1,000. So it's limited. But the good thing is that by the time lots of people claim they already do the observation of the microorganism, but they didn't record it, and he recorded it. You can see the recording. That's why people gave him a lot of contribution. But what happened is later on people found these are actually not bacteria. These are bacteria moving, rely on a special structure called flagella. So these are actually bacterial flagella moving. After moving, what they showed us. But it's good. He's the first person find the microorganisms. We will not be using those mechanical microorganisms, but in our lab, in the South Agriculture Science Building, we do have a microscope, which is a light microscope. And we will do a detailed introduction of the light microscope in our second day of the class on Thursday, okay? Louis Pasteur. This is a great German scientist, Louis Pasteur. Louis Pasteur have a swan neck flask experiments. It's a very interesting and a very famous one. The swan neck, which is from microbiology standpoint, repels spontaneous generation. It is a very interesting study. So you can see right here, it's clear. Okay, I have a container, let's say, a flask. Okay, I boil. Okay, there's no bacteria there. Okay, then I cut it. When you cut it, there are two different results comes out. One, you will see after they stay in the room temperature for a couple of hours, become muddy, become cloudy, which means bacteria is growing. Because the neck is broken. Then the other one is completely clear, transparent. It's sterilized, there's no bacteria there, or no microorganisms there. Now what's the difference? Because of this neck, the swan neck. The theory is that the bacteria has been hiding there instead of going to the broth. So that tells us the microorganisms, they will not generate spontaneously. Otherwise, this bottle will also become turbidity. So the bacteria contaminated the broth is because the swan neck is broken and the bacteria coming from the air. Okay, so that's a very magic experimental design to tell us to repel the spontaneous generation in the microbiology standpoint. Now, of course, people are going to repeat this study. They get different results. Some people find this guy is also become turbidity. Why? There are two reasons. Number one, some of the bacteria are heat resistant. They like the heat. They can survive the heat. So think about, did you ever go Yellowstone National Park at Wyoming? In the very hot springs, you still see bacteria there. There's a microorganism. Yes, those are thermal resistant bacteria. So they can survive. Another bacteria they can survive with the heat is endospore generation bacteria. Let's say Clostridium. You heard about. So in West Virginia, if you have a canned food, we have this regulation for that. We need to maintain the pH. The pH of the canned food has to be below 4.0 in order to prevent Clostridium botulism. So Clostridium is an endospore forming bacteria. That's endospore. Okay? So, this is two great scientists, John Tidal and Ferdinand Cohen. One of them find heat-resistant bacteria, and the Ferdinand Cohen find endospore generated bacteria, also heat-resistant. This is the final blow of repel the spontaneous generation theory. Okay, so that's a very interesting. Think about that. You're gonna get a different results. One cloudy, one not cloudy. When you do the research, what calls research? What means repeated. So people from different world will repeat it. They get different results, and they're gonna be mad. Oh, you say this is clean. How come there's a bacteria there? Then the people need to find the reason behind that. That's what John Tidal and Ferdinand Cohen they did. They find the reason is because heat-resistant bacteria could be, and they have endospore form. It's a very interesting topic. We talk about that. Robert Koch. This we'll talk on Thursday. He did a great work for the scientific area. Okay. I said it's gonna snow, so I will be off a little early today.